All right. Thank you so much for the introduction and the invitation to physically be here in Seoul again. It's one of my favorite cities to visit in the world. And so, yeah, I'm happy to be here. All right. So I'm going to talk about some new work that, in the sense that it's just published, but actually some really old work in the lab. We've been thinking about this problem for quite a while. Which is how to do actual statistical inference from single-cell RNA sequencing data. Here are my disclosures. So I think for this audience, hope all of you, especially with the talks preceding this, can appreciate that single-cell RNA sequencing throughput has continued to increase exponentially. And a lot of that has to do with experimental technology development and innovations. And some of it also has to do with these sort of like tricks that we've been able to come up with to try to get more cells into a particular experiment. And so this is sort of a set of methods that my lab published and also helped with in a few other papers where we basically are trying to sort of, game the 10x system, where instead of capturing a single cell within a droplet, we're encapsulating doublets as well. And if you have samples that are labeled, in our case, samples from genetically unrelated individuals, then you can very reliably detect these doublets and then also increase the number of singlets that you get out per experiment. And so, these approaches have sort of like now been demonstrated over and over again. And there was a talk earlier from UC San Diego about utilizing multiplexing approaches to increase the throughput of your single cell sequencing experiments. It also sort of, has other implications where we can generate better data. It's properly randomized for case control status or any covariates that you're interested in. And so, these approaches has really allowed us to scale single cell sequencing even more. And, I think now it's fairly routine that in an individual lab, you can sequence hundreds of thousands to millions of cells per experiment. And so I sort of see this as a major opportunity for using single-cell sequencing as an approach to study various mechanisms. And I think most of us are still doing single-cell sequencing to kind of identify within a complex cell mixture the various different cells that's within that mixture. But increasingly, we can also try to induce variation by perturbing the cells with genetic perturbations using CRISPR-Cas9 or utilizing natural genetic variants across different individuals. And so here's, for example, a study that we completed a few years ago where we used genetic multiplexing to sequence about 1.2 million PBMCs from about 200 different donors. This is actually a comparison of patients with lupus, which is a systemic autoimmune disease, with some healthy controls. In total, we sequenced something about 5,000 cells per donor. So we showed in that paper that if you sort of do genetic multiplexing and analyze your data carefully, single-cell RNA sequencing can actually be very quantitative in several different respects. But the easiest way to sort of think about this is to look at cellular composition, right? So what is the frequency of the various different cell types in circulation? And, we actually compare this to existing, just complete blood count data, and it was very highly correlated, and which gave us a lot of confidence to make these estimates that in lupus patients of both East Asian and European ancestry, there is this lymphodepletion that's explained by the loss of naive CD4 T cells. And it's sort of a very robust signal. So that's changes in composition, but what about, changes in cell state? So given a particular cell type, is there a difference between individuals with a disease versus healthy individuals? And so I think this is where existing methods really are quite limited. And, my lab is as guilty as any other to try to sort of make sense of the data, often just by using exploratory analysis. So for example, when we first published this paper, we looked at the distribution of cells from patients, which is on the right, and then healthy controls. You can kind of see that within the monocytes, which are these blue population, it seems like the patient cells are in a slightly different region than the cells from healthy controls, right? So we can sort of claim that this is different. Maybe we can subcluster this and look for genes that are differentially expressed. But I sort of would argue that this is all kind of hacky, right? We're basically doing statistical inference by just looking at the data. But statistical inference is something that, of course, has been around for hundreds of years, and so the key sort of thing that we actually want to do is hypothesis testing, right? So and what we really care about are questions around moments of distributions. And what do I mean by this? So I think most of us, when we think about any kind of sort of molecular quantification data, in the back of our mind, we're probably thinking about something like this: that you have a particular gene, and you have two populations of cells, and that the gene is probably normally distributed, at least if you think about protein expression. And so you can sort of look at these two populations and you can go, well, the average expression of these two populations are different. So that's the mean, right? So that's one of the moments of these distributions. But what's really cool with single cell sequencing is you can estimate other moments. For example, you can have a gene that actually has no difference between these two populations in terms of the mean, but the spread is different, right? In this black population, there's more variability. And so we can quantify this with statistical variance. And when you do single-cell RNA sequencing, when you're measuring 30,000 parameters in a single experiment, you can also do pairwise correlations between genes. So gene 1 and gene 2 could be correlated in this group of cells in black, and then not correlated in this group of cells in gray. And in terms of the covariates, we could look at differences between cell types, which a lot of us are already doing, or case control status, which is the example I gave earlier. But perhaps even more difficult is when we start thinking about these genetic perturbations, either natural genetic differences between individuals or genetic perturbations that we introduce into cells. And these effects are often extremely small, where you can't just cluster data and identify groups of cells that separate out by genetic variation or induced genome engineering. So what's the problem, right? So if this is the reality on the left, what happens when you do single-cell RNA sequencing is that you're sampling very inefficiently transcripts within a cell. And so even if your transcripts are normally distributed, which one could argue that they're not, when you do reverse transcription within a cell, it's a hypergeometric sampling process. And in fact, even with the best estimates of sampling efficiency, which is anywhere between 5 to maybe 20%, a distribution on the left basically looks like a distribution on the right here. And so, yeah, you can kind of tell that these two population cells have a different mean, but you can no longer just visually looking at this tell that there's a difference in the variance or correlation. And this is fairly representative of real single-cell RNA sequencing data, right? It's discrete. And it's even a bigger problem because we almost never sequence a library to saturation. We usually only sequence it to a certain amount that we can afford, and this is now a binomial sampling process. And so that's sort of like the fundamental problem. And so what do we do, right? And so what we do is if we want to compare differences in gene expression between populations of cells, we do pseudo bulk because the data is so sparse. And so in this data set of lupus patients, we could do something like this, where we pretend as though we had done a bulk RNA sequencing experiment and can identify the differences between cases and controls. The cases are red and the controls are in blue. And this is if you aggregated all of the cell types together. But of course, with single-cell RNA sequencing, we can cluster first and look at individual cell types, which is great. And so we can identify, for example, that these classical and non-classical monocytes have a different set of genes that are differentially expressed compared to the rest of the cell populations. And so that's sort of like, taking pseudo bulks of individual cell types that we identify in single-cell RNA sequencing, but we're leaving a lot on the table because ultimately we're not really using the entire data set, right? We're not using all of those replicate samples of monocytes within each individual as true biological replicates. And in fact, this paper that Square et al. Published now 3 years ago sort of really highlighted this problem, that if you looked at matched bulk RNA sequencing data with single-cell RNA sequencing data and ask, how good do existing methods for differential expression analysis do in recapitulating sort of a differential expression result from bulk RNA sequencing of sorted cells? It turns out pseudo bulk approaches, where you pretend your single-cell RNA sequencing data is bulk, performs better than all of these methods that's been developed for actually analyzing your single-cell RNA sequencing data, right? And so this is sort of a major problem. And so, we had a number of ideas of how to possibly solve this. And really what made it all happen was an individual graduate student in my lab named Min-Cho Kim. He's a MD PhD student in my lab, but was trained in statistics and electrical engineering. And so he was extremely quantitative and thought about the problem and came up with sort of the following observation. And I think I can't click next anymore. Let's go here. Here we go. And so what he sort of observed is that single-cell RNA sequencing, actually he proved this, can be modeled as I mentioned earlier, a hypergeometric sampling process. And what that is that you can think about all of the transcripts within a cell, and it's estimated anywhere from 200,000 to 250,000 per cell. And that when you're doing reverse transcription and having a probe to bind an mRNA molecule and convert that into cDNA, that's sampling without replacement, right? Once you've sampled that transcript, it's no longer there for other molecules to sample. And then the binomial sampling process of sequencing can also be modeled together with the sampling process of reverse transcription as a single hypergeometric parameter. And once sort of you have that, what you can do is we don't make any assumptions about the distribution of transcripts within a cell, because some genes are highly expressed, like housekeeping genes, and they'll look normal. Other genes, like transcription factors, are very bursty, and they could look quite sparse. So we don't want to make any general sort of assumptions about distributions of genes in cells. Instead, we just want to model what we're actually doing at the molecular biology step, which is hypergeometric. All right, so this Y is basically the actual counts that we would observe, and then we sort of like normalize it by some quantity because of hypergeometric sampling. And that will essentially allow us to get estimates of the mean and then the variance and correlation between genes. Okay, so we basically take the observed data and recompute the estimates for the actual data. Okay, so that's great, but there's some problems with the hypergeometric. Most of these problems are related to the fact that we can't easily calculate likelihoods on hypergeometric or derive asymptotics with the sampling process. And so whenever you have something like this, you have to basically utilize an approach called bootstrapping or shuffling the data, right? And so why has anybody done this? It's because whenever you think about shuffling data from single-cell RNA sequencing, especially when you get to hundreds of thousands to millions of cells, it can become extremely computationally inefficient. And so Min-Cho ended up figuring out how to do this extremely fast. So let me first talk about the estimators. And so the way that we really prove that this particular approach can work, and from a purely statistical perspective, hypergeometric estimators can be biased, which means that the actual estimate could be a little bit farther away than the real estimate. And so we wanted to make sure that it's not so biased that we're not reproducing the ground truth. And what we did is we analyzed a data set published by Arjun Raj's lab, where they did both single-cell RNA sequencing with DropSeq, as well as small molecule FISH, right? So this is in situ hybridization of the same cells. And so what we can do is then use Memento, which is the method, and see how well are the Memento estimates for mean, variability, and gene correlation correlate with the same estimates on the FISH data, okay? And so what you can see is that for all of these parameters, the Memento estimates perform much better than existing estimators, including for both variability and gene correlation, a number of these imputation-based methods, which borrows information from neighboring cells. So just by modeling the actual sampling process, you can get better estimates compared to FISH without any imputation of the actual data. Okay, so those are the estimators. What about the shuffling? So the key insight in this, and this is something I think is still quite striking in my mind, is that, it turns out just single-cell RNA sequencing data is extremely sparse. And so just how sparse is it? If you have, and this is a real PBMC data set, if you have something on the order of 30,000 cells and you have a matrix of 20,000 genes by 30,000 cells, the number of unique entries in that matrix that corresponds to a unique integer is on the order of 800, okay? So there's only 800 unique numbers in that matrix. But because of that observation, you can design a shuffling strategy based on the frequency of observing a certain count and not the actual columns of your matrix. And this basically approximate shuffling strategy will reproduce the actual shuffling, but at, about 1,000th of the time that it would take. And so allow us to basically do very quick shuffling for a single gene. So for example, even if you have 100,000 cells, just to shuffle all the data for a single gene will take you about 3 seconds. And so actually 0.03 seconds. And so you can now imagine parallelizing this across, a multi-threaded CPU to get shuffling times that are really, really fast. And so on average, if you're doing something like 50,000 or 100,000 cells, we can shuffle all of the data in 5 to 10 minutes on a single CPU. Okay, so what are the implications of sort of a better estimator and a ability to now calculate the confidence interval of that estimate? Well, the good news is that you no longer actually have to use pseudobulk anymore. And so here's Memento applied to the exact same data set that Square et al. Analyzed. And these two other approaches are also single cell-based differential expression analysis methods. And you can see that Memento is now first in class and beats out the pseudobulk methods on average. And it turns out some of these data sets have their own problems. And so we also analyzed an internal data set. Actually, it was the same as that lupus data set I showed earlier. What we generated is that for each of those samples that we did single RNA sequencing on, we also isolated 5 different cell types from the same cells and did bulk RNA sequencing on all of those cells as well across 200 people. And so, and you can see that in that data set, which is in many ways even cleaner, Memento does even better than the pseudobulk methods. Okay, so that's on differential mean analysis. What you can also do though with Memento is differential variance and differential correlation. So I just want to kind of walk through 4 different applications of Memento and show you guys some of the things that you can sort of use the method for. So this is a data set we generated during COVID, where we took some human bronchial epithelial cells that's grown on an air-liquid interface and stimulated them with both type 1, type 2, and type 3 cytokines, type 3 interferons, and did a longitudinal response, looking at the response of these cells at 0, 3, 6, 9, 24, and 40 hours after stimulation. And we're going to focus just on these ciliated cells since they're the most potent responders to type 1 interferons and also are implicated in COVID. So one of the things, the first thing you can do is you can use Memento and do differential mean analysis. And we can show, as we would expect, that alpha and beta interferons basically have very similar effect sizes on average expression of genes. And that's because both of them are type 1 interferons. And you can see that alpha and gamma are quite different because this is a type 2 interferon and this is a type 1 interferon. And then alpha and lambda are actually quite well correlated, but the type 3 interferon induces less of a response than a type 1 interferon. So this is all sort of known biology. And just shows that you can use the method to do differential mean expression analysis, and we sort of already know this. What you can also do is estimate variance, and this is something that's really interesting that we sort of discovered in the data set. And what we're now showing is the change in variability between cases and controls. So these are the changing variability in response to beta interferon versus the control cells. And so it turns out genes that reduce variability are some of the ones that are most overexpressed in response to the interferon on average, right? So this was kind of perplexing when we first saw this. In fact, this is an inverse correlation. But when you really think about it, it actually makes a lot of sense. So it turns out that some of these interferon responsive genes, when the cells are not being stimulated at all, are some of the most variable genes in the genome. But when you stimulate the cells with a cytokine, they all synchronize in their response. And this is in some ways how cells synchronize to each other, that we got a pathogen and we all should be producing this particular cytokine that neutralizes the pathogen, right? So this nice inverse correlation. You don't observe this nearly as much for gamma interferon, okay? So the other thing we can now do is differential correlation analysis. In fact, this actually further illustrates what I was saying earlier, is that these purple dots in the middle are all of the genes that are highly variable across all of the cells, even at baseline. And so in response to perturbation, they become less variable, but these genes are already co-expressed even in cells that are not stimulated with any cytokines. But in response to cytokine, as you can see that this changes dynamically over time, especially at 6 hours and 9 hours, it seems like there's a bunch of other genes that are now co-expressed with that core module of interferon-stimulated genes over time, and the co-expression goes away by 48 hours, right? So this is now a rewiring of a gene regulatory network dynamically over time. And you can sort of identify these relationships only by doing differential correlation analysis. So the second application is a data set that we generated internally where we sequenced 170,000 CD4 T cells. But what's unique about these cells is that they've also been perturbed. So these cells are edited at 280 locations in the genome, corresponding to about 140 transcription factors. And we did single-cell RNA sequencing all of this. So this is done with CropSeq. And these 140 transcription factors are chosen because they either are highly expressed in response to activation in CD4 T cells, or because the regulatory elements that these transcription factors are thought to be binding to are highly active in CD4 T cells, as estimated by bulk ATAC-seq. And so you can see that on the right, overall, the experiment worked. And this is Gen 1 of the CRISPR technologies in T cells. So it's surprising that it worked as well as it did. But there's this nice blue diagonal, right? So cells that are cells where we have a particular guide RNA expressed, if you look at the particular target gene, those cells will express lower levels of that target gene. So this is nice positive control that the experiment worked. But here's the kind of analysis you can do. So if you just do differential mean analysis, you can get something on the left, where basically each column is an average expression profile for each cell that has a particular guide RNA in it. And then each row is the average expression of all of those cells. And so you can see that, there's some of these really interesting regions that kind of light up. And so, for example, this cytoskeleton proliferation genes are turned on when these particular guide RNAs are introduced in cells, but so are these autoimmune Th1 and Th2 differentiation genes. And so you really can't tell that there's a difference between these two modules of genes if you just look at the average changing expression in response to perturbation. But now, if you look at the gene-by-gene correlation matrix, and this is a gene-by-gene correlation matrix made in cells that were not perturbed. So these are wild-type cells. You can see that it's actually quite different, right? These two modules of genes. These genes up here, the Th1, Th2 differentiation genes, are correlated with each other. And then this module of cytoskeleton proliferation genes are correlated with each other strongly. There's some cross-correlation between genes, but you can see that the clustering informs us of the differences between these gene modules. And so adding this additional layer of correlation can help us further interpret and map gene regulatory networks in response to perturbation. Something else we can do is now actually combine the differential mean and differential correlation analysis to detect epistatic interactions. So if you just look at the differential expression analysis results, basically what we can generate is a map like this, where each perturbation on a particular regulator, either R1 or R2, if it changes expression of the gene on average, we can sort of draw a line, an edge between the regulator and the target gene, okay? And so this is sort of a nice network, but it doesn't really tell you anything about causality or potential interactions between regulatory elements. But you can also do something like this, right? Because you have single-cell RNA sequencing data on the entire transcriptome, what we can basically ask is, if we knock out regulator 1, does it change the correlation of regulator 2 with the target gene? If the answer is yes, then we can hypothesize that these two regulators may interact with each other. If the answer is no, then these two regulators potentially interact independently to regulate this particular gene. And so we can identify, these candidate epistatic interactions, and we were able to validate some of them. So the third application is, we'll go back to that lupus data set that we generated. This is a data set that was collected on 200 some individuals, and each of these individuals were genotyped. So now we can look at the genetic control of these various gene expression distribution parameters. So the first thing, of course, is we can look at its effect on mean differences in gene expression. And what we're able to show is that this is basically mapping basic EQTLs in each cell type, is that Memento has increased power compared to even a pseudobulk-based approach. But there are things that you just can't do with pseudobulk, which is to map QTLs that control variability, as well as QTLs that control co-expression. And I'll show two examples of this. But let me just first talk about the mean QTL result. So what does this power increase allow us to do? Well, as sort of Tim mentioned earlier, one of the things that we were really excited about when we mapped QTLs is that the hypothesis is that if there's a genetic variant that's associated with gene expression, right, we expect that this particular variant could be modulating or perturbing a chromatin accessibility region in a particular cell type. And so if you do the pseudobulk analysis, you can see that there is this diagonal where EQTLs that are active in B cells are enriched for regions of open chromatin only detected in B cells. And that's true for T cells as well as CD8 and NK cells, which are pretty similar. But now if you look at the Memento results, this enrichment is much, much stronger, right? That there's much more cell type specificity for QTLs that we detect in B cells enriched for regions of open chromatin. What we can also do is, basically look at the rank sum statistics of explaining particular GWAS associations. And again, Memento produces much higher enrichment in each of these cell types compared to pseudobulk. And so I'll just give you two examples of some new sort of quantitative traits that we're still trying to figure out what they really mean. For example, here's a genetic variant that's associated with not just the average expression of HLA, but the spread of expression. So you can see that individuals who are homozygous for the A allele have much wider distribution for this particular gene compared to individuals with the G allele and heterozygous individuals. So this could potentially hint at epistatic interactions between this particular variant and some other gene. We can also identify co-expression QTLs. So this particular variant that's actually located in a cis-regulatory element of this gene LYZ controls the correlation between this transcription factor GUNB and the expression of LYZ. Okay, so individuals with the G allele have no correlation. Individuals with the A allele have great correlation. This particular case was easy to explain because that cis-regulatory, that SNP, that single nucleotide polymorphism, is located in a GUNB binding site, right? And so we have lots of examples of this. And so, we can't always explain it as a transcription factor binding a cis-regulatory element where there's a SNP in it. But this is sort of a nice explanation for why there's no correlation. And so as a last example, what we can also do with Memento is that shuffling approach that I described can be sped up even more. And so what we can basically do is go to a census, like the cell by gene discover census, which currently has about 100 million cells in it. And we can sort of regularly, maybe every couple months, shuffle all of the data, right? We don't want to do this every single time we want to do an analysis. We will just want to generate a null distribution whenever there's sort of a large update to the database. And when we have that null distribution, then we can power essentially arbitrary comparisons across multiple data sets. So this is actually our data in the cell by gene corpus, the lupus data set. And so you can, compare the different expression differences between CD4 and B cells, but you might also want to make comparisons between a lot of other cell types. And so you don't want to recompute your null statistic every time you do this operation. And so by pre-computing, this can be made much faster. It took a lot of engineering and in collaboration with the cell by gene team, but we show that a pre-computed p-value is very well correlated with the actual default p-value computed with Memento, which is already very fast. So this default is already using that fast approximate frequency-based shuffling. But this pre-computed one is even faster. And this is true for both mean and variability. And in fact, it's so fast to do the pre-compute that we can't even really, it's just essentially instantaneous. And so what that will allow us to eventually do is that you can go to a website and get near instantaneous report back of differential expression results. And so we test this out a little bit. So, why would this be useful? For example, we wanted to do a comparison between what are the genes that are differentially expressed between plasmacytoid dendritic cells and conventional dendritic cells. One of the reasons we want to do this is because PDCs in each data set is about 2%, right? So they're actually quite rare. And if you can combine across all the data sets where you sample this particular cell type, then you can improve your power. And so this is what we are able to show, is that the joint differential expression analysis with pre-computed Memento has better power, and that if you use each of these individual data sets, right, you'll have less power. Okay, so I'll just end with sort of like this video of what we hope to release. So a lot of the back end of this is already available. So many of you might have already played with this. You can basically go to the cell by gene, discover, pick up any arbitrary data set. So for example, this is our data set again, and you can choose sort of a random set of cell types that you want to compare. Actually, we're doing a case control comparison right now in a particular cell type. So you can take CD4 T cells, compare the expression of CD4 T cells between lupus patients and healthy controls. And it's fairly instantaneous right now, where you can get a result back. It's reporting out the T statistic results, which is computed with asymptotics, but we're working on the cell by gene team to also report this back out for Memento in the coming months. So we're pretty excited about delivering this capability to the community. All right, so I'll just end with the summary and sort of a few thoughts. So Memento is a new method for hypothesis testing for differences in mean, variability, and correlation from single-cell RNA sequencing data. Hope I've been able to convince you that it's accurate, it's robust, it's also super efficient, scalable. The details are described in the paper. We show an application of Memento to look at the response to stimulation, perturb-seq, and QTL mapping. And I think it's important to sort of like emphasize that statistical inference remains important and that methods matter. So I think there's a lot of exploratory analysis of single-cell RNA sequencing data, and there's also lots of prediction based on single-cell RNA sequencing data. But statistical inference remains, I think, one of the core sort of operations you want to do as a statistician to ultimately understand mechanism. And so let me just end with sort of this example, because I gave this talk actually at the NIH, and the person who spoke before me was the person who won the Nobel Prize for the Hubble telescope. And hearing the talk, it sort of reminded me of this, right? Like, when you look through a telescope and you see this, which kind of looks like a UMAP, by the way, how many stars are there, right? So if you just do inference using sort of visualization, you'll go, well, that looks like 5 clusters. I don't know what the mechanism is, but it's got to be 5 different cell types. But it turns out that there's only 2 cells, and or 2 stars, and this is an example of gravitational lensing, right? So the mechanism can produce a result that is not what we actually expect. And so, and by the way, I think it's also important to sort of take a look at this paper, which came out of Michael Jordan's lab, is that you can actually do inference with prediction methods, like deep learning methods as well, with some significant limitations, and they do a really good job of describing what those limitations are. All right, so with that, happy to take questions and acknowledge Mincho again, who single-handedly did all the work, and he's now in the ER being a real doctor. Thanks.